Hi, I'm Kevin Rychel, and I'm a PhD student in bioengineering at the University of California, San Diego, in Dr. Paulson's research lab. Essentially, what this paper is doing is taking a look at the Bacillus subtilis model organism, which is in soil and in your gut, and use machine learning to break down all of its gene expression into independent modules, which can be used to obtain new hypotheses about how the cell makes decisions and concisely summarize certain cellular differentiation processes. So it all starts with the transcriptional regulatory network, or TRN. This is the system that takes signal from the environment and converts it into gene expression. So traditional TRN methods include transcription factor knockouts, transcription factor overexpression, immunoprecipitation assays, and other wet lab methods which take a lot of effort and cost and require narrowing in on a single system. And the results that they get, while very important for gaining real mechanistic understanding, they're not very predictive of data. So we sought a different approach, which is independent component analysis. We take cells and expose them to a wide range of conditions, and then we measure their gene expression in each condition, and then we pass that into a machine learning program to try to understand the underlying structure. And this gives you a genome scale understanding of how the cell works. It's data-driven, and it requires minimal lab work in comparison to the other methods. This project started by downloading a transcriptomic data set from Nicholas et al. 2012, which is pictured as X on the left here. And we break that transcriptome down into imodulons, which are groups of independently modulated genes. And so each imodulon corresponds to a set of genes and a set of activities, which are the conditions in which those genes were needed. For example, our first imodulon was the fatty acid utilization imodulon, which contains all the genes needed for breaking down fat. It's active whenever the cell needs to utilize fat, for example, if the glucose is removed from its media. So this gives a pretty simple example of how we can look at both which genes are co-regulated and when they're needed. So throughout this paper, I break down the function of the 83 imodulons that were found, and we go through 5 hypotheses that came from curating these imodulons, as well as the process of sporulation and 3 new putative regulons. I can't discuss everything in the time for this video, but let's look at a few examples of how powerful imodulons can be. First, let's turn to the DNA damage imodulon. So this one is activated by mitomycin and peroxide, which makes sense because those supplements will damage DNA. But it's also active in swarming, which is this condition where you have a colony of cells and swarming cells swim away from it. And this is not something that has ever been related to, these two things have never been related in the past, but this result suggests that they should be. And if you look closely at the literature, you can see a potential mechanism, which is that DNA damage inhibits biofilm formation. And biofilm formation and swarming are sort of in this biological trade-off. And so we can expect that DNA damage might lead to swarming, and indeed that is what was found. Now let's turn to sporulation. This is the process where a normal cell experiences some sort of stress, such as amino acid starvation, and then it sporulates to produce a stress-resistant spore, which can then survive that stress and reactivate once conditions improve. And so what we've done is take the entire 8-hour process and break it down using our imodulons. And what we see is that the first hour is characterized by starvation response. So it tries to resynthesize all the amino acids that it can't find in its media. When that fails to prevent the starvation, then the cells go into a full community response where they stop growing, start producing toxins to fight away other bacteria, and take up cofactors that might be useful for sporulation. And then in the remaining hours, they actually sporulate. And our imodulon results also show the specific food sources that they use during this process. So this is a great example of how imodulons can be used to break down a complex process into something much simpler. Finally, one of the imodulons was related to this unique intercellular competition mechanism. So what you can see here is a recently discovered mechanism where the gene WAP-A is involved in protruding a fiber from Bacillus and using that fiber to attack an enemy species. This can then increase the amount of nutrients available to Bacillus. And so we found both additional genes that could be useful in this process, as well as the activity levels of this process. And we basically see that when the cell is starving, it sort of starts to hunt, and that under salt conditions in which membrane integrity might not be at its best, the cell will not use this process. Ultimately, what we found is that ICA can recapitulate existing knowledge and generate new hypotheses by exploring the activity levels and gene groupings that it produces. It also can be used to concisely summarize complex processes and is generally useful for anyone who wants to learn about the transcriptional regulatory network. If you're interested in this, please check out our paper in Nature Communications as well as our website, imodulondb.org. Thank you.